Hi everyone. Today's topic is male reproductive system, part 1. Let us discuss the topic under the following headings: Introduction, Functional and Anatomy of Testis, Spermatogenesis, Composition of Human Semen, Pathway of Semen. Male reproductive system consists of both the internal and the external genital organs. Let us discuss them one by one. Gonads, otherwise called as primary sex glands. Here in males it is a pair of testes, whereas in females it is a pair of ovaries. Accessory sex glands, seminal vesicle. There are two lobulated glands which is located on either side of the prostate. It is between the urinary bladder and the rectum. Prostate gland, it is the largest accessory gland of the male reproductive system. Bulbourethral gland or corpus gland, there are two pea-sized glands. They mucus-like secretion that enters the penile urethra during the intercourse. Ducts, ducts of the male reproductive system. Epididymis, it is formed by minute convulsions. Here it is. So they are tightly compacted that they appear very solid. Next is vas deferens, or otherwise it is called as ductus deferens. That is a continuation of the tail of the epididymis. Then ejaculatory ducts. So each ejaculatory duct is a slender tube that arises by joining the ductus deferens or the vas deferens with the duct of the seminal vesicle. Then urethra. So male urethra is a muscular tube, so that conveys the urine from the internal urethral orifice of the urinary bladder to the external urethral orifice at the tip of the glans penis. And then the supporting structures, they are the spermatic cord. It suspends the testis in the scrotum and contains the structure that pass through the inguinal canal, that is between towards the testis. In scrotum, it is a cutaneous fibromuscular sac which has testis, epididymis, and the lower ends of the spermatic cord. Then the penis, it is the male copulatory organ, and it is a common outlet for the urine and also the semen. Testis is the primary male reproductive organ. It corresponds with the ovaries in female. The main function of the testis is the production of the sperm, spermatogenesis, and the second one is they secrete the male hormone, that is the testosterone. So here in our today's class we are going to discuss about the male reproductive organs and also the production of the sperm. So testes, they are suspended outside the body cavity by the scrotum. Functional anatomy of the testes. Testes, they are the ovoid bodies which are suspended by the spermatic cords into the scrotum. So they are maintained at a temperature which is lower than the normal body temperature, which is very important for the normal spermatogenesis. So that is why the testis is lying outside our body to maintain the temperature, which has to be lower than the body temperature. So this lower temperature is very important for the spermatogenesis. And in an adult male, the average weight of each testis is around 25 grams. So the volume, around 90% of the volume of the testis, is because of the presence of seminiferous tubules. So each testis from inner to outer is covered by three layers. There are three layers. So the innermost layer is the tunica vasculosa. So this covering is made up of loose connective tissues. They are rich in the blood vessels. And the second one is the tunica albuginea, which is the middle layer. It is also called as the fibrous capsule of the testis. It consists of closely packed collagen fibers intermingling with many elastic fibers. This tunica albuginea on the posterior part of the testis, it expands into a thick mass which is called as mediastinal testis. So there are numerous septa from this mediastinal testis which project into the tunica albuginea, and thus they divide the substance of the testis into large number of lobules. And the third layer is the tunica vaginalis, which is the outermost covering composed of the mesothelial cells. The tunica vaginalis is a closed sac partially surrounding the testis. So there are two layers in it. The inner visceral layer. So this inner layer they adhere to the tunica albuginea, and the outer parietal layer of the tunica vaginalis, which lies in close contact with the inner surface of the scrotum. There is a small space between these two layers. So this cavity like structure has a small amount of fluid present in it. So covering of the testis provide protection from the trauma, and also it allows free movement of the testis in the scrotum. The arterial blood supply to the testis is mainly by the testicular arteries which arise from the abdominal aorta. This testicular artery, one of its branches, the anastomose with the artery of the vas deferens. The venous blood supply, the venous blood they are drained by the testicular veins which emerges from the testis and the epididymis and join to form a venous network which is called as pampiniform plexus. They consist of 8 to 12 veins lying anterior to the vas deferens, and they surround the testicular artery in the spermatic cord. This pampiniform plexus is it plays one of the important role in the thermoregulation, which maintains a constant temperature that is lower than the normal body temperature. Here there is a countercurrent mechanism which plays an important role. If anybody asks where is the countercurrent mechanism in our body other than the GIT and the kidney, you can say this is mainly by the pampiniform plexus. And the lymphatic drainage. So drainage of the testis happens to the lumbar and the pre-aortic lymph nodes. Then innervation, the autonomic nerves of the testis arise as testicular plexus of nerves on the testicular artery, so which contains vagal parasympathetic fibers and sympathetic fibers from T7 segment of the spinal cord. Testis, each lobule of the testis consists of seminiferous tubular compartment and the interstitial compartment. The seminiferous tubule compartment of each lobule of the testis contains about 2 to 3 seminiferous tubules. The seminiferous tubules constitute about 90% of the total testicular volume. This seminiferous tubule, each of them is around 80 centimeters long. It consists of two parts. One is the convoluted part and another is the straight part. The convoluted part forms the loops and they continues as the straight ends. So near the apex of the lobules, the straight part they ends and they join one another to form 20 to 30 larger straight tubules. This straight tubule they pass through the fibrous tissue of the mediastinal testis and they unite to form a network which is called as the rete testis. At the upper end of each testis, this rete testis gives 10 to 20 efferent ductules which continue into the head of the epididymis. And finally, the interstitial compartment. So it is the space between the seminiferous tubules which constitute around 10% volume of the testis. This compartment of each lobule is connected by the loose connective tissue. It has blood vessels, lymphatics, and also the Leydig cells. These Leydig cells have the most important endocrine function of secreting the testosterone, which we are going to discuss in our next class. Here you can see the epididymis. So this is the lobule and the convoluted one and the straight one forming the rete testis, and here is the efferent ductules reaching and forming the epididymis. So we can see where is what. Seminiferous tubules, they has three layers. The outer capsule or tunica propria, they consist of fibroelastic connective tissue containing few muscle-like myoid cells. So the contraction of this myoid cells, they help in the movement of the spermatozoa along the walls of the seminiferous tubules. And the next one is the basement membrane or basal lamina. It is a thin homogeneous lamina which lies next to the outer capsule layer. And then epithelial layer, it is a complex stratified epithelium. So they are arranged in two compartments. One is the basal compartment, another one is the abdominal compartment. So here this epithelium consists of mainly two types of cell. One is the spermatogenic cells, another one is the substancular cells or centralized cells. Here you can see the seminiferous tubule, and here you can see the primary spermatocytes. Here you can see the spermatids, and this is the centralized cells or substancular cells. And here you can see the matured spermatozoa. Here you can see the view of the electron microscope. This is how the seminiferous tubule is arranged. Spermatogenic cells, they lie in between the centralized cells or the substancular cells. All these cells are spermatogenic cells. This is the centralized cells or the substancular cells. So they are arranged in an orderly manner in 4 to 8 layers, which extends from the basal lamina to the lumen of the seminiferous tubules. In children, this testis is not fully developed, so only primitive germ cells, that is the Spermatogonia, is present in the children. In sexually mature individuals, the spermatogenic cells of all the stages, so in different stage of differentiation, that can be seen, and they will be always arranged in an orderly manner. The basal compartment or the deep part of the epithelium is occupied by the cells of early stages of spermatogenesis, that is the Spermatogonia and the primary spermatocytes. The abdominal compartment, so it contains cells of later stages of the spermatogenesis, that is from the periphery to the lumen. These cells are secondary spermatocytes, and then the early and late spermatids, and also the spermatozoa. Centralized cells, they appear as irregular pyramidal shape. Centralized cells occupies both the basal and also the abdominal compartments. Tight junctions they form as there is no cytoplasmic continuity between the two adjacent centralized cells, but the plasma membranes of two adjoining cells are connected by the tight junctions. Here you can see this is a tight junction. In the basal region, so it is present. So the wall of the seminiferous tubule is divided into two compartments. So the tight junction forms effective permeability barrier within the seminiferous epithelium, which is also called as a blood testis barrier. So this blood testis barrier, it limits the transport of many substances from the blood to the seminiferous lumen. These barriers, they maintain the germ cells, that is sex cells, in a privileged location, because mature sperms are very immunogenic when they are put into the systemic circulation. So this barrier protects the cells of different stages of spermatogenesis from the toxic substances which is present in the blood, and also from the antibodies. So this layer also prevents the entry of the byproducts of the gametogenesis into the blood. Sometimes the breakdown of this barrier can lead to the immunogenic response against the germ cells. The maintenance of the luminal fluid composition happens, so which contains glucose and protein in a very low amount when compared to the plasma, and the testosterone and the potassium in high concentration. Functions of the Centralized cells. These cells, they provide the physical support to the maturing germ cells, and also they nourish them, so as it has more glycogen products, and also they remove the waste products from the germ cells. Phagocytic function, there are residual cytoplasmic organelles which are cast off from the spermatozoa during the conversion of spermatids to sperms. So these cytoplasmic residues are phagocytized by the Centralized cells. Secretory functions. So Centralized cells, they secrete Mullerian duct inhibitory substance, MIS, or what is also called as Mullerian duct inhibition factor, which is secreted by the Centralized cells. So this MIS, which help in the regression of the Mullerian duct in the fetus, only by which it can keep the male sexual development. And then inhibin, so it is a substance that inhibits the spermatogenesis before the onset of the puberty by regulating the FSH secretion. And then transport proteins such as transferins, which transports iron, and ceruloplasmin, which binds with the copper. So these transport proteins are formed by the Centralized cells. And then plasminogen activator, it is required for the proteolytic activities, for the disruption of the tight junction during the migration of maturing germ cells from the basal to the luminal compartment. So this plasminogen activator is formed by the Centralized cells. And then estrogen, so small amount of estrogen is secreted by the testes. There is an enzyme which converts testosterone to the estrogen, which is done by the aromatase enzyme. So this aromatase enzyme is formed by the Centralized cells. And then seminiferous tubular fluids. So these Centralized cells, they secrete the watery solute rich fluid into the lumen of seminiferous tubules. This fluid movement provides a driving force for the non-motile spermatozoa. And they also secrete the androgen binding protein, which is secreted by the Centralized cells. It has high affinity for the testosterone and also for the hydroxytestosterone, as they maintain the high concentration of androgens in the lumen of the seminiferous tubules. Epididymis is 7 meter long which is formed by minute convolutions of the duct of epididymis. So they are so tightly compacted and they appear solid. So the head part, which is the superior part, they receive the spermatozoa. Body, the vessel in the inferior portion, and the tail, they lead on to the ductus deferens. So the efferent ductus, they transport the sperm from the retatus to the epididymis and they get stored there. The sperms, they remain viable for the month in the epididymis. The secretion of the epididymis, they provide the nutrition to the spermatozoa and they also help to mature. The non-motile spermatozoa become motile only when they pass through the epididymis. So the main function is the storage of the sperm and the maturation of the spermatozoa by making it motile. Vas differences or ductus differences, that is the continuation of the tail of the epididymis. It ends by joining to the duct of seminal vesicle. It serves as a secondary storehouse for spermatozoa, which will be released at the time of the ejaculation. In vasectomy surgery for sterilization in males, the vas difference is ligated and they are sectioned. So the main function of the vas difference is to transfer the sperm to the urethra. Seminal vesicles, there are pair of seminal vesicle present. They do not store sperms. They secrete a thick alkaline fluid that mixes with the sperms as they pass into the ejaculatory ducts and the urethra. The duct of each seminal vesicle joins the vas deferens to form the ejaculatory duct. So the seminal vesicle fluid, they have fructose for the energy and the prostaglandin. So they are secreted from the seminal vesicles. The seminal vesicle produce 60% of alkaline semen. So that includes fructose, which gives energy for the sperm. Bulbourethral glands or copper glands. It is a pea-sized pad at the base of the penis. They produce around 10% of the semen. So alkaline mucus, like secretion that enters the anterior urethra during the sexual arousal. So they give protection from acidic pH in the vagina when the spermatozoa enters after the intercourse. So the main function is to secrete the mucus and the alkaline buffers to neutralize acidic conditions of the urethra. Prostate gland is the largest accessory gland of the male reproductive system. They maintain the alkalinity and the clotting enzymes from the prostate. So the main function is they produce up to 30% of the semen, and it includes the nutrients and also the enzymes to activate the sperm. Urethra, they transport urine from the urinary bladder, and sperm and the semen produced by the male reproductive organs. During the emission phase of ejaculation, the sperm are entered into the urethra by the sympathetically induced contraction. The motor neuron induced contractions of the skeletal muscles at the base of the penis, they expel the semen during the expulsion phase of the ejaculation. So the main function of the urethra is to transport the urine and the semen. Spermatic cord, they, they suspend the testes in the scrotum and they contain the structures that pass through the inguinal canal, so which goes into the testes and comes out of it, and it contains the vas deferens, the blood vessels, and also the nerves of the testes. The coverings of the internal spermatic fascia, premasteric fascia, and the external spermatic fascia. Spermatic cord can be palpated as it passes over the pubic brim. It constitutes the pampiniform plexus of the spermatic vein, spermatic artery, and then the vas deferens and the lymphatic vessels and the nerves, that is the iliolingual and genitofemoral nerves. Scrotum is where the testes are located, and its function is to provide the environment for the testes, which is below the normal body temperature. So this lower temperature is very important and necessary for the production of viable sperm within the testes, and its main function is to support and protect the testes and to maintain the temperature of the testes, that is approximately below the normal body temperature. The structure is the skin and underlying superficial fascia, dartos muscles in the dermis, and cremaster muscle, which is continuous with the abdominal wall muscle. So involuntary contraction of the scrotum can happen, that is a cremasteric reflex. So the scrotal sac that forms two separate chambers, so the right and the left one, by the septum of the scrotum. Penis is an organ for the discharge of urine and for the deposition of sperm into the vagina during the copulation. Penis can be divided into three parts: the root, which is fixed to the ischial ramus, and then the body, which is where the erectile tissues are present, and then the glans penis, that is the enlargement of the corpus spongiosum. So there is prepuce in the glans penis, which is a foreskin partially that covers the glands and that surrounds the external urethral meatus. So this part, the prepuce, can be removed during the circumcision procedure. And then the prepucial glands, which produce the smegma. And then the main function of the penis is the urination and the copulation. So penis is composed of three cylindrical bodies of erectile cavernosus tissue, the corpora cavernosa, and then the corpus spongiosum. So these structure is very important for the erection, which will be discussed during the male sexual act. Book Spermatogenesis. It is a process of formation of spermatozoa from the Spermatogonia, that is a primitive germ cell. The characteristic features of the spermatogenesis, it begins at puberty and continues throughout the adult life, which declines in the mid old age. The majority of the Spermatogonia, the primitive germ cell, they undergo the continuous mitotic division to provide the additional stem cell. Only a minority of the Spermatogonia undergoes further differentiation by meiosis, a process which is unique to the germinal epithelium. So each differentiating spermatogonium develops into 512 spermatids. Spermatogenesis does not occur simultaneously in all parts of the testis. At any one moment, some areas of the seminiferous tubules are active, while other areas will be in the resting state. In humans, it takes at an average of 74 days to form a mature sperm from a spermatogonia. The spermatozoa formed in the seminiferous tubules are non-motile structures. The spermatozoa then undergo the process of maturation while passing through the male genital tract, and they acquire the ability of fertilizing an ovum. Spermatogenesis happens in different phases. The first phase is the phase of mitotic division of spermatogonia. The spermatogonia, or the primitive germ cell, serves as a pool of undifferentiated stem cells from which develop the spermatozoa. So these cells, they lie near the basal lamina of the seminiferous tubules. Three main types of spermatogonia are there. First one is dark type A or AT, also called as type A1. So they represent reserve resting stem cells. They have dark staining oval nuclei. They divide to form more dark type A cells and also some light type A cells. And the second one is light type A or AP spermatogonia. They have light staining oval nucleus. They divide to form more light type A spermatogonia and also some spermatogonia of type B. And the type B spermatogonia has spherical nucleus. Each type of spermatogonium divides mitotically 5 times to form 32 type B spermatogonia. The division occurs in the basal compartment of the seminiferous tubules due to incomplete cytokinesis. All cells derived from the single spermatogonium remain connected throughout the cytoplasmic ridges. So the connections remain throughout till the formation of the individual spermatozoa. And the phase of formation of primitive primary spermatocytes by mitotic division. The 32 type B Spermatogonia undergoes mitosis to form 64 primary spermatocytes. So the primary spermatocytes are large cells with large nucleus having diploid number of chromosomes, that is 2n. Phase of formation of secondary spermatocyte by meiotic division. After the first reduction division of the meiosis, the 128 primary spermatocytes forms 256 secondary spermatocytes having haploid number of chromosomes, that is 23. Where the size of the secondary spermatocyte is quite small when compared to the primary spermatocytes. And then the phase of secondary is a phase of formation of spermatid. Each secondary spermatocyte, they divide into second meiotic division, and they give rise to two spermatids from one secondary spermatocytes. So there will be a total of 512 spermatids formed from a single spermatogonium. The early spermatid is a small round cell with spherical nucleus containing haploid number of chromosomes, that is 23. The late spermatids are formed when the early spermatids undergo changes in the shape and orientation of the organelles. The phase of formation of spermatozoon or the spermiogenesis. Here the late spermatids, they do not divide further, but they undergo some morphological changes and they form the sperms. The spermatids, they undergo changes in the form of shape and orientation of its organelles. Spermiogenesis. So during the formation of the spermatozoon, the components of the spermatids which they take part in the forming the spermatozoon, they undergo some changes in the nucleus, undergoes condensation and changes in its shape to form the head of the spermatozoa. So the nuclear proteins are replaced by protamine which surround the chromatins, and thus they protect from the mutagenic factors. The centriole divides into two parts. The distal part forms the axial filament, and the proxial part, they form the basal body. In the beginning, both these parts lie in very close to each other, but later they move away. The region earlier occupied by the centriole, later later on they form the neck of the spermatozoa, and then the Golgi apparatus, they form cap-like structure covering the anterior two-thirds of the head, which is the acrosome. They contain this Golgi apparatus. They contain the proteolytic enzyme hyaluronidase, and other proteases which help the sperm in the penetration of the womb during the fertilization. And then a mitochondria, they surround the tail part of the sperm, lying wrapped in a sheath, and they provide the energy for the movement of the spermatozoa. Cell membrane, cell membrane of the spermatid consists as covering of the spermatozoa. The membranes of the late spermatids and they contain the germinal angiotensin converting enzyme. A fully formed spermatozoon is a motile cells which has 50 to 65 micrometer in length. The structure of the spermatozoon. So the head, it is oval in shape. So mainly they are composed of condensed nucleus with a very thin cytoplasmic cell membrane layer around it. So it is surrounded by a acrosome, which is a thick cap-like structure which covers the anterior two-third part of the head. It it is formed mainly from the lysosome-like organelle containing a number of enzymes like hyaluronidase, proteolytic enzymes, and acid phosphatase, which helps the spermatozoa in penetrating the ovum during the fertilization, as we have discussed earlier. The next one is the neck. It is a narrow constricted part which contains a funnel shaped basal body and a spherical centriole. This basal body is also called as the connecting piece because it helps to establish an intimate union between the head and the remaining portion of the spermatozoa. The basal body is made up of 9 segmented rod-like structure which become continuous with the axial filaments which is present in the tail. Tail. So the tail of the sperm is the motile portion. It is also called as flagellum. It is divided into 3 parts. The first one is the middle piece, then the principal piece, and finally the end piece. Axoneme or the axial filament, it forms the central skeleton of the tail. The axoneme, it begins just behind the neck and extends through the entire length of the tail. So this structure is similar to the cilia. So the to and fro movement of the tail, it results due to the longitudinal sliding motion. And then the coarse fibrils, they are present in the middle piece and the principal piece, but they do not extend into the end piece of the tail. And then the fibrous sheath is present outside the coarse fibrils, and then a mitochondria. So in the middle piece region, the proximal part of the tail, the fibrous sheath is surrounded by spirally arranged mitochondria, which is abundance. This mitochondria, they synthesize the ATP, which supplies the energy for the motility of the tail. Storage of spermatozoa. Around 120 million sperms are produced each day. A small quantity is stored in the epididymis, and most of them are stored in the vas deferens and also in the ampulla of the vas deferens. They remain stored, maintaining their fertility status for about a month. Maturation and capacitation. The role of epididymis. Fully developed spermatozoa are released into the lumen of the seminiferous tubules. From there they reach the epididymis after passing through the rete testis and the efferent ducts of the testis. The epididymis is the site of extratesticular maturation of the spermatozoa. When the sperm arrive in the epididymis, they are non-motile. They acquire this motility feature only after passing through the epididymis. But this epididymal secretion, which is important for the maturation of the spermatozoa, are androgen dependent. The role of seminal vesicles and the prostate gland. Secretions of the seminal vesicles and the prostate, they have a stimulating effect on the sperm motility, but the spermatozoa become fully motile only after this ejaculation. Role of female genital tract. When the spermatozoa is introduced into the vagina, the spermatozoa reach the uterine tubes much sooner than their own motility would allow. So this suggests that the contraction of the uterine and tubal musculature, they exert the sucking effect. Capacitation. So spermatozoa, they acquire the ability to fertilize the ovum only after they have been in the female genital tract for some time, that is for an hour to 10 hours. So this final step in the maturation is called as a capacitation. Factors affecting the spermatogenesis. Temperature, effect of temperature. The proper spermatogenesis occurs at a temperature lower than the normal body temperature. So location of the testis outside the body cavity in the scrotum, and the evaporative cooling effect in the scrotum, the countercurrent heat exchanger mechanism operating between the spermatic artery and the vein, so where the Companiform plexus plays an important role. And then there are evidences which suggest the importance of the proper temperature maintenance during the spermatogenesis. Hot bath at a temperature of around 43 to 50 degrees Celsius for 30 minutes daily, they can result in the sterility. And cryptorchidism, it is a condition in which testes are undescended. So in such individuals, usually they become sterile because of the um, defect in the thermal regulation, and they can damage the spermatogonia. And then varicocele, it is the dilatation of the vein of the Pampiniform plexus. So the varicocele occurs due to the incompetence of the venous walls, so which results in the retrograde blood flow. In that condition, the heat exchanger mechanism is completely uh, disturbed, and that can cause the sterility. And then the seasonal variation in sperm count can happen. So they have observed the sperm count is greater in the winter season than in the summer season. So irrespective of the temperature for which the scrotum is exposed. And then the infectious diseases. So like mumps, they can cause the degenerative degeneration of the seminiferous tubules, so that can decrease the spermatogenesis. And then the hormonal control that we can go through in our next class, the GnRH control. Semen or the seminal fluid, it refers to the fluid which is ejaculated during orgasm at the time of the male sexual act. The average volume of the semen per ejaculation is 2.5 to 3.5 milliliter after an abstinence of 2 days. So the volume of the semen, it can decrease with the repeated ejaculation. Vas difference, they contribute around 10% of the volume. Seminal vesicles, they contribute 60%. Prostate gland, they constitute 30% of the volume of the semen. The pH is 7.5. There is fibrinogen which helps for the coagulum that dissolves after 15 to 30 minutes because of the fibrinolysin which comes from the prostate gland. The lifespan of the sperm in the vagina is 1 to 2 days. So in the low temperature, it can maintain several weeks. At minus 100 degrees Celsius, you can freeze it for years. So now they collect the semen and they store it, and that can be stored in the semen bank at the temperature of minus 100 degrees Celsius for several years. Composition of human semen. So the color is white and opalescent. Specific gravity is 1.028. pH is 7.5. The sperm count. So the normal sperm count, they vary from 35 million to 200 million per ml of semen, with an average of 100 million per ml. So 50% or more with a count of 200 millions per ml, and essential of all those which counts under 20 million per ml, they are considered as sterile. So although we need only one sperm to fertilize an ovum, so even 20 million per ml is considered as sterile. And then the secretions of the seminal vesicles, they contribute around 60% of the semen volume. So the secretions from the seminal vesicles is mucoid and viscous fluid. They are neutral and slightly alkaline in nature. They contain fructose, phosphorylcholine, ergothioneine, ascorbic acid, flavins, and prostaglandins. So the main function of the seminal vesicle secretions are to give nutrition to the sperms So by the fructose and other nutritive substances, and the clotting of the semen, which happens soon after the ejaculation, is mainly because of the fibrinogen, and fertilization of the ovum. So they may be enhanced by the prostaglandins. Composition of semen, the secretion of the prostate gland. So the prostate gland secretion contains spermin, citric acid, cholesterol, phospholipids, fibrinolysins and fibrinogenase, zinc, acyl phosphatase, calcium phosphatase, bicarbonate and hyaluronidase. So the prostate secretion contributes 30% of the total volume of the semen. So they contribute milky and alkaline fluid part of the semen. So the maintenance of the optimum pH, which is required for the fertilization, is the function of the alkaline prostate fluid. So this neutralizes the acidity of the vaginal secretion. So at this pH, the spermatozoa become motile and the chances of the fertilizations, they are enhanced. Lysis of the coagulum, this is caused by the fibrinolysins, which is present in the prostostatic secretion. The sperms, they become motile after the lysis of the coagulum. And then there are secretions from the bulbourethral gland and other mucous gland. They account only a very small amount of the semen volume. These secretions, they provide the mucoid consistency to the sperm after the puberty. The pathway of the sperm: seminiferous tubules, retate testis, epididymis, vas deferens, ampulla of vas deferens, ejaculatory duct, prostatic urethra, membranous urethra, and then finally the penile urethra. In summary, the spermatogenesis, which is the process of formation of spermatozoa from the spermatogonia. So there are 5 phases of spermatogenesis. So the first one is the phase of mitotic division of spermatogonia. So each spermatogonium divides mitotically 5 times to form 32 spermatogonia. And the phase 2 is the formation of primary spermatocytes by the cell division. The 32 spermatogonia, which has 46 chromosomes, undergo cell divisions to form 64 primary spermatocytes. And the primary spermatocytes are the large cells which with the large nucleus having the diploid number of chromosomes. The third phase is the formation of secondary spermatocytes by the meiotic division. So each primary spermatocyte undergoes the cell division. So after the first division of meiosis, the 64 primary spermatocytes are converted into 128 primary spermatocytes with 46 chromosomes, diploid number of chromosome. The 128 primary spermatozoids, they undergo the cell division to form 256 secondary spermatocytes having haploid number of chromosomes. So the fourth phase is formation of the spermatids. Each secondary spermatozoa divides mitotically to give rise to the two spermatids. So a total of 512 spermatids are formed from a single spermatogonium. And then finally the formation of spermatozoon, that is a spermiogenesis. The spermatids, they do not divide further, but they undergo the morphological changes to form the sperms. The spermatids, they undergo changes in the form of shape and the maturation. To read in the Purana, Gaitan and Hall, Genome, and the GK Paul. Thank you.